Hello, everybody. Okay. To many in the audience today, our speaker requires no introduction. Travis Sawyer is a newly minted assistant professor in the College of Optical Sciences. He's also an assistant professor in health sciences, an assistant research professor of medicine, and a member of the UA Cancer Center and the UA Bio 5 Institute. So while Travis is in some sense our newest pandemic baby, he actually interviewed for this job in the spring of 2020. He's actually, as many of you, also an elder statesman here at the OSC. He received his PhD from our department in 2021 in Jennifer Barton's group, and his BA from the OSC in 2017. In between, he received a master's degree from Cambridge, and in the meantime, he received, I think, pretty much every award that the OSC has to offer, so I don't have to list them. So the important thing is we'll learn today is that Travis is doing cutting-edge research in biomedical imaging. His new laboratory, the Biomedical Optics and Optical Measurement Lab, explores in particular cancer detection, including development of screening devices incorporating optical coherence tomography, fluorescence imaging, and other novel imaging modalities which focus on image analysis through machine learning techniques. So prior to his faculty life, Travis started a company developing visual recognition software for detailed image capture, which enabled discoveries in astronomy and art preservation in addition to medical optics. Travis is also a strong supporter of science outreach, regularly organizing and supporting efforts to educate the community and encourage careers in science. So one interesting thing to note here is that 3 weeks ago when I approached Travis to give this talk at the last moment, he said, actually, I'm in the middle of an oral surgery. I'm told I can't eat solid food, but I can talk, and this is a perfect opportunity to prepare for a colloquium. So you can't me to keep a good man down. So with that, thank you very much, Travis.
Yeah, thank you, Dal. I'm really excited to be here today, and I clearly can talk all right. I was a little nervous last Thursday, but it all turned out okay. As an aside, I definitely wouldn't recommend that you get your salivary gland removed if you can avoid it. But in any case, I'm really happy to be here today because, again, I've been on the other side of this desk many times, and this is the first time that I'm on this side in my new role. So today I'm going to be talking a bit about the new research that my lab has been doing. I'm going to cover a bunch of the different projects that we have going. But the main premise of what I'm talking about is this concept that I refer to as a spectral fingerprint. And so I titled the presentation, Identifying the Spectral Fingerprint of Disease Using Optical Imaging to Shed Light on Cancer. And so I'll talk a little bit more about what I mean by that specifically and how we can use optical imaging to really advance healthcare in terms of diagnosis and screening. So to set the stage a little bit in terms of the motivation, we first have to understand a bit about cancer and what it means to detect it and screen it. So I'm mostly going to be talking about epithelial cancers, and so that we might have heard the term carcinoma, but essentially the epithelium is the surface layer of the tissue, and so all of these cancers begin on that initial surface layer. We see that they're some of the most deadly and the most common cancers in the world. For example, the 5 that I'm listing up here are essentially the 5 most common organ systems to develop cancer. And we see that the vast majority of them are carcinomas. If we look at the number of deaths that these carcinomas cause, we can see it's quite high. It's about 250,000 a year in the US alone. Of course, if you extend that to the world, these trends continue. But the main idea is that carcinomas are deadly, they're common, and so as a result, we really want to try and reduce the burden on the healthcare system caused by them. And so as I said, epithelial cancers, what does it mean to be an epithelial cancer? It means that it begins in this surface epithelium, which begins sort of right at the surface of the tissue. So here I'm showing a schematic of an esophagus, and so it's kind of a tube of tissue. So the epithelium is that inner layer. So it's the thing that's most kind of out to the world. For example, your skin, you have epithelial tissue sort of on the outside as well. So this makes it an excellent paradigm for optical imaging because when we're trying to image things, what we're doing is forming an image of a flat surface or just a surface in general. And so things like esophageal cancer or other GI cancers you can access using something like an endoscope, take optical images, and use it for screening and diagnosis. It really sort of pushes that minimally invasive paradigm. And so in general, epithelial cancers are great for optical imaging, but you really need to motivate specifically why do we need to detect this early and what does early detection do for helping patient outcomes. And so again, here I'm just showing esophageal cancer, but these trends are similar for pretty much all epithelial cancers. And we see that this is really a key example for the need for effective screening. And there's kind of two facets to this. The first is the question of how well are we doing in terms of detecting it early? And the answer is not very well. We can see that in this pie chart, I'm showing this number of cases or the proportion that are diagnosed at a particular stage. And so about 40% are diagnosed when they're in the distant stage, and so this is when they've already metastasized and spread to other organ systems. About 30% would be diagnosed in what's called the regional stage, and so here it's begun to spread locally, but it's not yet left that organ system. And then only about 20% are diagnosed when it's in the localized stage, and so this is still when it's just a single lesion that's growing in the esophagus. So that answers the first question of, do we have very effective early detection? The answer is no. And the second question is, what does early detection give you? Well, we can look at the 5-year survival rate for each of these stages of diagnosis. And so when it's diagnosed in the distant stage, the survival rate is extremely low, just about 5%. It's extremely deadly disease. If we detect it early in the localized stage, we increase that survival rate by about 10-fold, so almost up to 50%. Still not great, but it's significantly better than 5%. And so clearly, if we had better early detection, we would have higher patient survival, better patient outcomes, not to mention that the treatment for these varies significantly depending on what stage it's at. Localized, it's very easy to do a local resection, whereas with distant spread, you have to have an esophagectomy where your entire esophagus is removed. Terrible quality of life afterward. And so overall, getting better early detection and screening can really improve patient outcomes. It can save lives. And this is true of many epithelial cancers. I'm just focusing a bit on esophageal cancer since my main area of interest is esophageal and just general gastrointestinal. And so today what I'm going to talk about in the presentation is a couple of different topics. And this kind of follows the development of how you might create a new screening or detection technique. So you start off with what are called characterization studies, where you essentially get a bunch of samples of normal tissue, of diseased tissue. You can test out different modalities, for example, fluorescence imaging, or I'll also talk about multi-photon imaging. And you figure out which image features change the most between normal and diseased tissue. This then becomes kind of the motivation for, okay, now we need to build a classifier using this data. So you might introduce some algorithms, whether it's machine learning or statistical based, that can help you classify whether the images that you get are showing healthy or diseased tissue. And then ultimately you need to develop the devices to acquire this data in vivo, in the clinic. And so that's kind of the progression I'm going to follow today, as I'll talk a bit about some of our optical imaging characterization studies to try and identify different imaging features that can help us diagnose things. I'm going to talk some about the projects involved with algorithms and machine learning, and then I'll conclude talking about device development, so building devices in terms of new endoscopes or new imagers that can help us with screening and diagnosis. And so starting off with the optical imaging characterization, I'm first going to go a little bit into depth into what I mean by the spectral fingerprint of tissue or of disease. And so when we talk about optics, we of course have light, which can be different frequencies or wavelengths, and we have that on the electromagnetic spectrum. Here I'm just showing a quick diagram of the visible spectrum where it goes from violet to red. And so what you see is different biological mechanisms will give rise to light being emitted in different portions of the spectrum, and it actually generates a very unique signature. So for example, if you excite the tissue down in the blue or violet wavelengths, you can actually probe things that are related to the cell structure, for example, the amount of connective tissue and whatnot. And so this, you could say, is important for cancer because as you see the cancer proliferating, you see that the cell structure becomes more and more disordered, gets very randomized, and so the amount of fluorescence in this region actually changes quite a bit. So I would call that cell biology you could probe. Up in the red, you are very sensitive to things like blood and the concentration of oxygen within that blood. And so within the context of cancer, what happens is that you have these cells growing and growing, and their proliferation is really not bounded. They grow a bunch of blood vessels to bring more oxygen and sustain that increased energy production. This is a hallmark of cancer. It's called angiogenesis, but we see that this causes a lot more blood to be present in those regions. Interestingly, kind of in contrast, is that as this tumor grows, it gets bigger and bigger. And so the center of it is actually further from the blood vessels that are around the periphery. So you see that the core of that tumor actually begins to decrease in oxygen. It becomes hypoxic. And so by measuring different levels of blood oxygenation, you can actually see that it has this kind of core which begins to die off or even go into this different type of energy production not using oxygen. And so using imaging to probe oxygen levels and blood concentration can be very effective for cancer diagnosis. So I would call that probing the cell metabolism. And then finally, there are various fluorescent proteins and amino acids all across the visible spectrum that you can try and detect depending on which wavelengths you illuminate with and detect with. And so I would call that the underlying chemistry. And so the main idea with this is that as cancer progresses, these properties of the biology, the chemistry, and the metabolism are going to vary significantly. And this gives rise to different light-tissue interaction, which gives it a unique spectral signature. So this is what I call the spectral fingerprint, and the idea is that by measuring these, you can differentiate between diseased and healthy tissue. So more specifically, what is it that we can actually measure with different types of optical imaging? Of course, that's a very broad term. So if we're measuring something like the reflectance and scattering characteristics, this of course is sensitive to different types of molecules, different sizes, and different densities. And so that could be things like cell organelles, like mitochondria, or of course just the transition between two materials like fat and muscle. Similarly, absorption is a strong optical characteristic of tissue, and that's almost entirely dominated by some major absorbers like water, hemoglobin, and so that also depends on how oxygenated the blood is, but hemoglobin is present in blood. And then also lipids, so fats. So those are the main absorbers. If you probe more coherent properties of light, like phase and polarization, you can get information about specific molecules that have polarizing effects. And so one of the most common ones is the connective protein collagen, and so that pretty much holds everything together, and that causes a strong polarizing effect on the tissue. But you can also have circular polarizations as well. For example, glucose. Glucose is present a lot in the body, specifically your brain or in your gut, and that produces a chiral effect that has circular retardance to it. You can have nonlinear effects, and so I'll be talking a bit about this later in terms of multi-photon microscopy. But here, essentially, you can have two photons that are absorbed or interact with a molecule simultaneously. Then you have one photon coming out on the other side. So that would be called a frequency doubling effect, but you can have frequency tripling, as well as Raman effects as well. And then, of course, most of us probably are familiar with fluorescence, where you essentially have some molecule that absorbs light of a certain energy. Eventually, it will re-emit light of a different color after some energy loss. And so depending on what that molecule is and what the underlying energy structures look like, you'll have different conversions between excitation and emission. So we'll talk a lot about fluorescence as well. And then finally, you can even get into things that convert light to sound waves, which is called the photoacoustic effect. But I won't be talking about that today. So starting off, we're going to go into some of these characterization studies we're doing. And so specifically, we're using a lot of multi-photon and fluorescence imaging for this. And so both of these can be explained in a very similar way using just these standard energy diagrams that we've probably all seen a little bit. So whenever you have a molecule, you have energy states, and normally your electrons are sitting down in the ground state. At some point, you can introduce energy into the system. In this case, it's light, which gets absorbed, and it promotes those electrons up to the higher energy states. Over time, you have vibrations, which cause the little bit of energy loss, but then eventually you'll have the electron returning to the ground state, and you have the light re-emitted of a slightly longer wavelength, and so slightly lower energy because you lost some to those vibrations. So this is standard fluorescence. This can happen both naturally. There's various things in tissue that cause fluorescence. Some of those prominent are related to the metabolism. So a couple things I'll talk about are called NADH and FAD. These are just two molecules that occur a lot with the metabolic processes in cells. And so if there's a lot of NADH, there's a lot of metabolic activity, a lot of energy. Similarly, you can have artificially introduced fluorophores. And so that could be through a dye. You could spray it on the tissue. That might bind to a certain receptor. You can introduce it through IV, or you can actually genetically engineer DNA to produce fluorescent proteins. So I'll talk a bit about that later, but that tends to be less acceptable in terms of diagnostics, given that you have to worry about toxicity and whatnot. So in terms of multi-photon, we have a very similar picture where you can have two photons being absorbed to promote that electron up to the energy level. Same idea. You have some vibrations, relax it, and then it re-emits later of a different color. So we would just call this two-photon fluorescence. Again, it's wavelength specific. But then we have a really interesting effect called second harmonic generation. And so in this case, it is a scattering event, and so it's nearly simultaneous, whereas fluorescence has a lifetime of, on the nanosecond scale. This is more on the femtosecond scale. But you have two photons which interact with a molecule. I wouldn't really call it absorbed, but they almost immediately are re-emitted of exactly half, or exactly double the energy in a single photon. So here there's no energy loss due to vibrations, and this is only produced by molecules of certain symmetries. And so we see in tissue the most significant is, again, this connective tissue collagen. And so if we look at the maps that we can get of tissue of these, what we see is that when we look at multi-photon images, we're getting an idea of the local variations in the cell metabolism, as well as the connective tissue and its integrity. So on the left here, this is a 2-photon fluorescence image, and these big, bright depositions tend to be of, as I said, very metabolically active tissues, or tissues that might be very, they express this product called lipofusion, which is related to when the cell is undergoing oxidative stress, which is, again, when it runs out of oxygen. And so that gets produced significantly when you have cancerous tissues. And so you can see that there are various local regions here that are quite bright. I don't have scale bars on this. This is just meant for qualitative visualization, but this is microscopic. So this field of view is probably 500 microns by 500 microns. On the right here, this is the second harmonic image of collagen, and you can see that you get the really nice mapping of the extracellular matrix. So looking at things like how randomized that is particularly relevant for cancer diagnosis. And so in the study that we're doing, we're applying multi-photon imaging to try and identify and detect these spectral fingerprints of a particular GI cancer that occurs in the small intestine. And so the system that we're using, I just have a nice little diagram up here, that we actually have a tunable titanium sapphire laser. And so we're doing this at multiple different wavelengths to try and specifically target different naturally occurring fluorescent markers. So I mentioned NADH is a metabolic cofactor. Porphyrin is related to blood. Lipofusion, again, is related to this oxidative stress caused by the lack of oxygen. SHG here, this is just second harmonic. And then FAD, again, is a metabolic cofactor. So we select different excitation wavelengths. We have it pass through the system, goes through a beam splitter that lets it hit the sample, goes through lots of optics for focusing. And then here at the sample, we're imaging. We have 15 human patient samples of this gastrointestinal cancer called gastronoma. And then we're also imaging normal tissues, which the closest thing to normal you can have is this gland in the body called the Brunner's gland. And so I'll show images of that. But basically, we have our normal and our diseased tissues. Then, of course, on the return, it travels back through, passes through the beam splitter, because now that the frequency has been doubled, it no longer gets hit by that long pass filter, or rather, it gets redirected by the long pass filter. It goes through a secondary beam splitter, and it goes into this really nice detector that you can vary different spectral channels for detection. And so essentially, it has a series of diffraction gratings that you can rotate at different angles to let yourself capture really whatever spectral range you want on the return light. And so we are detecting specific wavelength bands for each of these biomarkers as well. So I'm just showing a couple of images of what we've collected. So this is an image of a normal Brunner's gland. The left, this pinkish image, this is what people would normally look at in biology. You stain it with two chemicals that are abbreviated H and E, where one of them binds to the cell nucleus, it makes it a purple color. The other binds to the cytoplasm, makes it a pink color. And so this is kind of the standard of care in terms of diagnosis. But what I'm zooming in here is a transition point between normal epithelial tissue, so again, that's like the surface, and then the Brunner's glands that are underneath it. What I'm showing in the other two columns are our image of the NADH channel and of the second harmonic channel. And so you can see really interesting image features. For example, in the NADH channel, you can see that there's a lot more metabolic activity in the epithelium, which is expected because that has really high turnover rate. And then the Brunner's glands have less, and they've also got these kind of open areas, since these end up being essentially tubes that you're taking a cross-section of. If you look at the second harmonic, that gives you a really nice mapping of the overall connective tissue that holds everything together. And so that these images tend to be really effective for kind of mapping out how things are oriented and whether they become disorganized. So this is normal tissue. This looks fine. And then we imaged a tumor. And so again, this is just one example. We have the H&E slide up here, the NADH, and then the SHG. And so interestingly, you can kind of see, there's a tiny little Brunner's gland right next to the tumor here, but you can see that the tumor is just this massive clump of metabolically active cells, and so it's all quite bright in the NADH channel, whereas the Brunner's gland, again, has these voids and whatnot. But then also you can see that it really has it's almost compartmentalized in this larger sort of area of collagen here, where we have kind of a large boundary delineating the tumor area compared to the normal tissue, which is very different from the Brunner's glands, which has the tiny little tubes and stuff going on. And so clearly there's quite a bit of different metabolic and structural activity going on with these tissues. And so we've done the imaging on these, and the next step is to actually craft imaging features to try and identify the differences in the areas between healthy and diseased. And so one of the most interesting, in my opinion, is when you take the ratio of NADH and FAD, that tells you a little bit about how much, again, of the metabolism is being produced by the oxidative pathway, so the way that it uses oxygen to produce metabolism, versus the pathway that does not use oxygen. And so that's related to that oxidative stress concept. So taking the ratio of those can tell you quite a lot about the underlying metabolism of the tissue. But then also, of course, we have structural characteristics that we want to measure, for example, the morphology. And then one of the main hallmarks of this cancer is what's called stromal thickening. So the stroma is the area in between. It's all of that connective tissue. And you can see that in the normal area, it's quite thin and wispy, whereas in the kind of abnormal area, it gets really, really thick. And so we're thinking that this is, there's almost a transition point between when the Brunner's glands becomes a tumor, where it gets really thick and kind of messy and stuff. And so as we go forward, we're essentially trying to craft these imaging features to really be diagnostically useful. So how else can we use fluorescence in multi-photon imaging? Well, in another way, we can use this in a wide field of view sense. And so that was all microscopic stuff, very small, micron scale resolution. We can also look at this as kind of a wide area screening approach. And so in this project, we're using fluorescence imaging. In this case, we're just collecting one fluorescent bandwidth, but we're using it to track what's called cell lineages. And so when you have a cell, you can label it with a particular fluorescent protein. Here we do it by editing it into the genome. But essentially, as that cell replicates and produces, it continues to express that fluorescence. And so we want to track how it proliferates through the body. And so in this case, this is a mouse model of colon cancer, where we've labeled the cancer cells with this fluorescent protein. On the left here, I'm showing a control, and we remove the organs after the animal is sacrificed. And so I'm showing the liver, the colon, and then the way that the GI tract goes is you have the duodenum right after the stomach, the jejunum after that, and then the ileum. And so you kind of have duodenum, jejunum, ileum, and then colon. And so clearly with the control mouse, we don't really have anything interesting that we're seeing. But when we image the transgenic mice, we see that we actually can do this lineage tracing, and they appear as these tiny little speckles in the organ. I don't know if the resolution on the screen is good enough, but you can sort of see that these speckles start to appear. And interestingly, with if you guys remember from like high school biology, you can have genes which are expressed in 2 pairs or 1 pair. And so when you have the case where the mouse is expressing the gene in both pairs, it actually has a lot higher abundance of these speckles. And so it's clearly showing that the burden of cancer cells is much higher. So the idea with this project is we're measuring it at different time points. This is just one time point. This was about 3 weeks after the mice had been sort of activated in terms of the fluorescent compound, but we're also seeing how quickly this proliferates in terms of 24 hours, 3 days, 7 days. And this tells us a lot about how the cancer actually can progress through the body, which could ultimately let you develop better therapeutics and better diagnostic approaches. So this is great, but of course one obvious drawback is that we have to take the organs out. That also kind of poses the problem of you can't measure the same sample at multiple time points because, fortunately, the mouse is no longer alive. And so applying this in vivo would be a lot more useful. For example, you could put the mouse to sleep. You might have some kind of imaging probe, which we could introduce through small incision. And you could collect fluorescent imaging to show cancer location, the tumor bulk. Maybe you have different locations where cancer is growing. But you could also maybe take a biopsy while you're doing that to get some cell analyses going, determine whether it's actually the type of tumor that you want, since there are many different subtypes, as well as doing things like genome analysis. And so in another project, we're developing a small imaging probe to do this, so we can measure these things in vivo. And so we have a mouse model that we've just started growing, where we're trying to develop this cancer that occurs in the small intestine. And so essentially, it's a bit arcane, but you take some cells in a syringe and you inject them into the mouse, and then the tumor begins to grow. And so we would call that a xenograft, but we have a xenograft model of this gastronoma, and so that's what I'm showing here. You can't see it very well, but in the first go of one of these things, you always just do it underneath the skin. You do a subcutaneous injection to see if it works. And so you can see that this actually is the tumor beginning to grow, this dark area. And so these, this cell line clearly is viable for this. And so we've developed this tiny little device so that we can actually monitor these things as they grow in vivo. So one of my students has designed this and is assembling it. And the idea is that you have a nice little holder that kind of fits everything together. We'd call that the ferrule. And then you have a couple of fibers for illumination. So you can couple in a fiber optic, or you can couple in a light source through those fiber optics for your excitation light. You have a miniature camera, which then has this sort of fancy multi-band filter in front of it. And so this thing we specifically picked so that it could let you do 4 different types of fluorescence imaging at not necessarily simultaneously, but kind of in series. You can just cycle through different illumination wavelengths and excite different fluorophores. And so you can see that the transmission of this filter, it dies off about 405, and so that's a common fluorescent wavelength, at 488, 561, and then 632. And so we can use those 4 different wavelengths to track up to 4 different labeled cells. So you can see this image here is just a initial prototype that she's put together. You can see that we've got some red light being coupled through it, and then the stuff's all pretty small. It's about millimeter by a millimeter. The whole scope is about 2.7 in diameter, but the actual components are just about a millimeter by a millimeter. So that kind of summarizes some of the characterization studies we're doing. And so now that we have a bunch of data, that's excellent. And of course, what we want to do is build up some algorithms and classification types of problems that we want to do. And so kind of just going back again to the basics of what are we looking at with reflectance and fluorescence. And I'm also going to start talking a bit about polarization here. But in terms of the signatures of reflectance and fluorescence, you have all sorts of things which are sort of encoded in the different tissues. And so on the left here, I'm showing the reflectance spectrum of different types of tissue. You can have colon, muscle, artery, vein, and they vary significantly, again, depending on what's composing that tissue. And then in terms of fluorescent, that's a little bit more specific. You have some very well-known fluorescent biomarkers like proteins, NADH, lipofuscin, and porphyrins. So when you're measuring any given spectral signature, it's going to be a combination of all of these things. And so it makes it quite challenging to actually try and identify what is creating that signature. You just get something that looks like a wavy line, and how do you go from that to say, okay, well, clearly that has higher metabolic activity, so it's cancer. It's quite a challenging process, both in terms of building up a model for this, but actually also building up a data set. And so one way to build up a nice data set for this is to use what's called hyperspectral imaging. And so hyperspectral imaging is similar to spectroscopy, but you're essentially collecting the full spectrum for every point on an image. And you can do this with various methods of scanning. You can either get it point by point. In this case, it literally is just doing spectroscopy for each point. You can collect for a full line and then collect the data with a 2-dimensional detector and scan line by line. You could scan through wavelength using something like a tunable filter or a filter wheel. And then they have these really fancy systems called snapshot systems, but in my experience, these tend to not be super robust. So usually you'll find it's a scanning method. And with hyperspectral imaging, you're collecting reflectance, absorption, and scattering information, as well as a contribution from fluorescence. And so again, it's all of this mixed up signal. And so what do you do with that? Well, you have to do what's called spectral unmixing. You have to kind of remove the different not remove, but rather differentiate the different components contributing to your signal. So a lot of this was pioneered in the scope of remote sensing, actually. They use a lot of hyperspectral imaging for doing things like assessing different types of land cover, whether it's forest or water. And the idea is that you have your signature, which is shown in a pixel here. It's all sort of messed up. And then you can unmix it into different end members, which are the components. And so you might have one portion of vegetation, one portion from the background signal, one portion from mineral, and then some man-made stuff. And so this is essentially the whole challenge with doing spectral imaging and trying to identify spectral fingerprints, is that you want to know what is it that's contributing to that unique spectral signature. And so in tissue, you would want to try and unmix things like the amount of collagen, the amount of porphyrin, the amount of NADH, and whatnot. So just tying it again back into the actual biology, you want to know things about the metabolism, about the structural nature of the tissue. but it's the same premise of trying to unmix that signal. And so when you have hyperspectral imaging, you can also have a subset of that called multispectral, where instead of collecting the spectrum at really high resolution, you're just collecting a couple of discrete wavelength bands within that, usually less than 10. And so multispectral imaging actually becomes quite popular for things that need to be done more rapidly. And so, for example, with tissue imaging or in the clinic, things need to be very fast, either because people don't stay still, or when you remove tissue, it degrades super quickly. And so it needs to be a couple minutes, because if it starts to degrade, then it kind of loses its purpose, and you can't actually do any useful analysis on it. And so multispectral imaging becomes the predominant form of biomedical imaging. And so one of the areas of research that I'm very interested in kind of working on, especially with collaborators in University of Cambridge, is to develop these custom developed multispectral filter arrays. And so these things are multispectral imagers, but the idea is that you have a sensor which has the filters patterned directly onto the pixels themselves. And so the most common form of this would just be our standard RGB camera, like in our phones, which is called the Bayer filter, where we have one red, one blue, and two greens. So that's over every 4 pixels, and then we just replicate that over and over and over. Then when you actually have light going through the system, it filters it out, and you have an image which is sort of checkerboarded in terms of your signal. And then to recover the full thing, you do something called demosaicing. But this is just the simple case of your RGB sensor. You can do this with arbitrary filters as well. And so, for example, this is a multispectral filter array. This particular mosaic design is 2 by 3, so it has 6 different filters, and you can see it's evenly spaced kind of across the visible spectrum. And so you can imagine that this could become quite useful, because when you're dealing with tissue, you actually see that the spectral information, you have a lot of redundancy. You don't necessarily need to collect the entire spectrum to be able to identify which of the components are contributing to it. And that's because these spectra are, 1, well known, and 2, they vary very slowly. They don't really have really sharp, narrow peaks. And so knowing that they vary slowly and that they have pretty well-known line shapes, you can actually do okay using multispectral compared to hyperspectral. So just showing a comparison up here, hyperspectral would be sampling most points, or as many points as you can on this wavelength spectrum. The two curves I'm showing up here are for hemoglobin and deoxyhemoglobin. So these are what you measure when you're trying to estimate oxygenation. And so you could collect all of it. That would do fine. You could probably get your measurement, but it would take a while, and it wouldn't be sustainable to something clinical. Not to mention that hyperspectral things can be a bit bulky. You could try doing RGB, but the fact is that those aren't specific. And so they just take some data, they mix everything up, and they really have no scientific basis for why. They have a scientific basis, but it's just for visual perception. You know, it's not for trying to unmix different tissue components. And so doing an RGB sensor isn't particularly useful. So the premise is that you could design these things to be optimized to pick the specific points on the spectrum which are most relevant, which let you then recover those individual end members at the highest accuracy. And so the idea is that you could develop custom multispectral filter arrays custom multispectral filter arrays tailored to detect these spectral signatures or spectral fingerprints. And so just one example I'm showing here is, again, RGB on the left, but then you might have one application which needs different spectral bands shown as these little vertical lines. Maybe your multispectral filter array looks like this. Similarly, you might just need one that has 6 bands. So that application might have a different concept, and this one might just be 4 bands, and so on. And so things like the number of bands, that becomes a question. Where they're positioned becomes a question. How wide is the bandwidth becomes a question. It's actually a very challenging optimization problem. And so designing these custom things, it's complex and takes into account multiple variables. For example, the spectral aspect. We've talked about that this whole time, where what wavelengths do you care about, what do you want to unmix, but you also care about spatial resolution. Those of you who are perceptive would notice that if you have more and more filters, you're going to have a lower effective spatial resolution because you're only having one filter every 3 or every 6 pixels. And so that becomes a problem as well. And so you have to worry about spectral selection. You have to worry about the spatial demosaicing process. You can recover some of the spatial resolution, but it's never perfect. And then, of course, the computational problem. How do you unmix your different components, and how do you classify different tissues? And so just this little facet that I'm showing up here, I'm not going into detail, but this is essentially a study we did with esophageal cancer, removing the tissues and trying to train a machine learning algorithm to classify it. So it takes into account all of these different variables, making it a really challenging optimization process. And so one of the kind of main areas that we're moving toward in this is developing this optimization framework for trying to tell us what is the best multispectral filter array, but also, after you have the data, what do you do with it? You know, it's going to be in this sort of spotty checkerboard form. How do you get it back to the best resolution image you can, and what do we do with that in terms of a classification model to try and tell us what we're looking at? And so in this model, essentially, you start off with your input data, which would be a hyperspectral cube. We call it a cube because it has two spatial dimensions and then a spectral dimension. And I'm just showing this as apples, but essentially this would be tissue data. You would have healthy and diseased. But then you would also have the ground truth classification. We would call that the end member abundance. So that could be something like your end member is cancer and your end member is healthy, or if you cared about oxygenation, it would be the two, deoxyhemoglobin and hemoglobin. So the idea is that with this input data set, you start off with a kind of initial conditions for your multispectral filter array. So this includes things like the number of bands, the filter response. It doesn't have to just be a Gaussian. It can be other shapes as well. Of course, the mosaic pattern, what arrangement it's in. And then also there's different methods for demosaicing, and so you have to pick different ones. So with this input and these initial conditions, you can simulate what your multispectral filter array would collect, and then try and reconstruct and classify what your object would be. You then would go back and look and see how close that is to your original ground truth end member abundance, and then adjust your parameters and continue to optimize. So it's just a standard optimization process, but it takes into all of these different takes into account all of these different variables. And so trying to put this to the test, we've tested it on a couple of different data sets for tissue. This one was some removed esophageal tissues, and we pinned them, and then we had a pathologist look at the tissue on a grid and tell us whether the different spots were healthy or diseased. And so you can see that here on the left we have one with a bunch of intramucosal cancer. There's a little bit of what's called Barrett's esophagus, which is like a precancerous condition, and then a little bit neoplastic. And so that's that early stage that you really care about. And then on the right, this is just an example of normal tissue where you have some with normal, and then a little bit with Barrett's, which may or may not eventually become cancer. So we had a bunch of samples of this. We input this into our algorithm where we used the spectral signatures and kind of came up with a, this is sort of a contrived scenario where we just put the center as cancerous, the ring as early cancerous, and then around the edges as normal. And we tried to figure out what's the best multispectral filter array for this and how well can it unmix. And so we tested out different types of classification. For example, this is using k-nearest neighbors. This is using a support vector machine. And then we did 3 filters, 4 filters, and 6 filters. And so something that you actually find is that more filters doesn't necessarily give you better performance. At some point, you've collected enough of the kind of information content that when you introduce more and more, it's just introducing noise into the system, and you actually start to see a decrease in the performance. And so in some cases, you get this sort of like kickback effect. And so really doing this optimization doesn't always tell you that having 100 filters is the best. And so in this case, we actually found that 3 filters using the k-nearest neighbors was the highest performing, or 4 using the support vector machine. We also tested this with some colon tissue that we collected the data for. So actually, I took this data a long time ago. I went up to Oregon Health Sciences University, and we took the data in just routine colonoscopies. But same premise. We took a bunch of images of polyps and cancer, a bunch of images of normal colon, and then we had our spectral signatures, kind of shown in this figure here. And then we came up with a contrived scenario saying, oh, okay, we've got some areas that were polyp, areas that are post-resection polyps, so after you've removed it, so clearly there's going to be lots of blood there. We have areas that are benign, and then areas that are just the specular reflection of it coming back into the system. And so that's the little circles there. And so here, again, we did the multispectral filter array optimization, and we found that really in all cases it performed very well at trying to identify those different components. And then we tried to go a step further, saying, can we determine the amount of oxygenation in that? And so that's kind of what these mappings are showing, where the top is the ground truth, and then the next 2 are different approaches for doing that with the 4-band multispectral filter array and the 6-band. So you can see it's not doing great, but one reason for this is because it was optimized to unmix the different types of tissue, not trying to unmix the different types of oxygenation. So again, that really kind of motivates the need for having particularly relevant knowledge beforehand of what you want to do and what this should be customized for. These aren't supposed to be something that's generalized to everything. They're supposed to be targeted for a specific purpose. And so where are we going with this? Well, so one thing that I'm particularly interested in is integrating different types of polarization sensitivity. So you can actually pattern on these little filters, little polarization filters as well, different types of linear polarization states. And so we're kind of trying to move that toward integrating that into the model. That would give us a bit more structural information, as well as integrating different kinds of imaging modalities and then things like genetic analyses. You know, genes tell us a lot about what could be giving rise to the mechanisms of cancer progressing, and so you can imagine that someone going into the clinic, they might go get imaged, but they could also have a gene panel done, and this could be combined in some kind of multimodal data synthesis. And so sort of having this idea of design and optimization, but then actual data analysis, where we're integrating in other types of modalities as well. But there's two main things that kind of arise from this, that your model or algorithm is only as good as the data that goes into it. If you have really bad training data, you're going to have terrible results. It's not going to be translatable. It's not going to be sustainable. And so this really motivates the need for building really specific and accurate characterization systems to collect your data. And then the other thing comes up of how do we fabricate these sensors? And those of you who are familiar with lithography might say, oh, well, I mean, this is great, but doing something customized and doing it, you know, doing a different one for each disease, that's not, you can't do that. That's not sustainable. And so this kind of brings me to the final part, which is about the device design. And so first I'll talk about that idea of building up a comprehensive data set. How is it that we can actually get good enough data to do this in a sustainable way? But then also, how can we fabricate these things and test them? And so as I said, to develop these things accurately, we really need to have robust and accurate data. And so this really all starts here, where you start with the reference hyperspectral data, and if you want polarization, you need to have polarization sensitivity in there too. And so that includes things like the reflectance of the tissue, the fluorescence, and then also the polarization sensitivity. And so we've seen these two figures before. Now I'm kind of adding in a little bit with polarization, but the idea is that this is showing the orientation of fast axis of the retardants, but that very much aligns with very well-ordered tissue structures and stuff. And so one of the projects that's ongoing is to build up a sort of a comprehensive tissue imager. This is in progress, so it's not quite complete, but the idea is that we have one section which will do multispectral fluorescence, and so we'll have a series of filters that get rotated through. We have a xenon light source that comes in, and we can filter out for excitation. But then in tandem, or rather in series, we could then collect some polarization-sensitive hyperspectral data. So this is a hyperspectral camera with a rotatable polarizer in front of it, so we can probe different polarization states and get the whole hyperspectral cube. So that really provides an ideal data set for generating these multispectral filter arrays for different diseases. And so just a little bit about the actual hyperspectral imager. I find it quite interesting, but as we talked about, you kind of need to scan. Could be scanning point by point or line by line. The way that they do it to make it fast and robust, so you don't have any registration issues, is you actually have a little filter on top of the sensor, not bonded to the top of it, but over it, and then you sweep the sensor underneath the filter. And so you're essentially scanning, but on the sensor side. And so here you can see that your object and the camera actually remain stationary, but this little thing, this little sensor inside gets swept from side to side, letting you collect every wavelength band for every point on the image. And then it reconstructs the cube. And so these filters work just through the Fabry-Perot principle, so different filter heights get you different transmission for different wavelengths. And so what this actually looks like is you have your filter array, which goes from red to blue. There's a section here with no filters that you need for doing accurate registration, and also tolerances in terms of the actual design of these things. But you can see that the quantum efficiency plot down here, you have a bunch of narrow band responses over the entire spectrum. And so this goes from about 450 to 900. And so you get both areas where fluorescence will be occurring, but then also a lot of the strong contributions from where blood has a strong signal. And so I find this really an innovative design. It makes it really fast to collect these things. It takes, it's less than a minute, essentially, where normally with hyperspectral data it can be minutes 10 or 15 minutes. A lot of my work in my master's was trying to do that with line scanning systems, and it just takes forever. So again, this operates through Fabry-Perot. So the idea is you might have a dielectric stack where you pick different thicknesses of the components on the stack. Some of them will give you constructive interference, depending on if the wavelength is perfectly reflected, or if it's off by half a period, you'll have destructive interference. And so the way that this looks physically is essentially it's a big staircase. You have a staircase of these filter things, and then you just sweep your sensor under it. And so this provides great preliminary data set for us to do the optimization, but finally we need to talk about how do we actually fabricate these things. If you want to develop them custom, you have to develop a fabrication method that can be adjusted to give you different center wavelengths, different bandwidths, different positions. All of these things are quite challenging to do with standard lithography. And so just covering the two methods that color filters are made. One, as I said, is a dielectric stack. It has to do with different filter heights. But then others, for more broadband, you can use color dyes. But for narrowband filters, you really need to do dielectric stacks. The dyes tend to not be particularly specific in terms of your wavelength filtering. And so with my collaborators, we've developed a method to produce arbitrary cavity heights on a lithography step, where we essentially vary the amount of energy exposure per pixel on a grid. And so, for example, what is the power behind this? Well, generally what you do with lithography is you have your object, you put a mask down, and then you expose it, and then you take it off. And then either the photoresist has remained if it's positive, or it goes away if it's negative. Then you wash off the rest, and you have something left over. In that case, you use uniform energy, so everything stays the same, or everything is deposited at the same amount. In this case, what we're doing is every step, every pixel, we essentially vary the amount of exposure dose, either leaving more of the photoresist behind, again, if it's positive, or if it's negative, you'll be getting rid of it. We've generally used positive, so it's essentially you produce more energy, leaves a higher filter height. And so what we're showing here in the top left is just a simulation that if you have a particular wavelength and you have a cavity height, the transmission is shown on this chart here. So red is really high transmission. So you can see sort of that at 450, we have really high transmission at 225, and so that makes sense because one round trip is 450 nanometers. And then, of course, we can convert that to the exposure dose needed to produce a given cavity height. And so that's what this plot is showing here, that we can vary the exposure dose to produce the cavity height to give us the transmission. So these are some of the example images that we've collected using scanning electron microscopy, where basically this is just making the staircase pattern, and then this one is doing like essentially ramps, ramps in the horizontal direction. And so we've tested this by actually monolithically integrating these onto a big silicon wafer. We cut this thing up, and then we bond it to the readout circuitry, and so we can see if it actually produces the spectral signatures that we want. And so that's kind of what I'm showing down here, where this was, again, just testing these things out, and our resulting transmission properties were fairly good. Bandwidth is around 15, 20 nanometers. But we have complete control over the central wavelength, and we can adjust the bandwidth to some extent as well. And so that lets us physically realize those optimization things that we've been doing. It's great to do something in a computer, but until you can build it in real life, it has limited utility. And so what we did with this was try and build one of these things to try and unmix oxygenation. And so again, that's pretty much always the first step that everyone always does in spectral imaging, is you see if you can get the hemoglobin, deoxyhemoglobin, and figure out how much oxygen's in it. So we built, we plugged in the data into our algorithm. We had it select the spectral filter array, patterned that onto a sensor, stuck it in a camera, took some images. And so these, again, are quite preliminary, but essentially it's showing that the capillaries in your finger have a lot more oxygen than the surrounding tissue, which is qualitatively what we would expect. As an aside that I won't go into, this is actually how you diagnose various skin conditions. You look at the capillaries, and they are only really looking at the structural characteristics of them now. So having some way to assess oxygenation is useful in a healthcare sense. So this is great. We've tested it preliminarily, but now we really want to bring this back to cancer. How do we get this into kind of a diagnostic sphere? And so we would want to build this into something like an endoscope. And so some of the current work is, again, trying to actually build this into an endoscope, which can couple into existing instruments, so we don't have to worry about things like FDA approvals. We can just stick it through the biopsy channel. But the idea is very similar to the device you saw earlier, that you have a bunch of fibers that are delivering your light. You might need to do some uniformity stuff with microlens arrays, but on the return, you have an imaging lens, which then goes right onto your multispectral filter. As I mentioned, we're moving toward polarization sensitivity as well. Whether, in some cases that may be useful, some cases it may not, but that's all part of the optimization problem. And so ultimately, the goal in this is to try and improve just diagnosis in terms of making it more rapid, cost-effective, and just more accurate in general. So the idea of how this might work operationally is that you'd have your physician doing a standard procedure. You could introduce this through what's called the working channel. You take your multispectral and polarization sensitive images, which are giving you information, as I said earlier, about things like the naturally occurring fluorescent biomarkers, as well as absorbers and whatnot, but then also the polarizing structures that have to do with the connective tissue. And so ultimately, that might be something that augments the standard of care. So they're doing their biopsy. We're not trying to replace these things, but rather give them an additional look. Maybe point out the areas where they might need to go biopsy later. Interestingly, with esophageal cancer, it's a complete random approach. They go in there, they take random biopsies in your esophagus, and you just hope. And so in this case, if we could make it more specific, more targeted, we could have that be a lot more sensitive and more successful. And so that brings me to the end. I, of course, just want to acknowledge my excellent lab group and the different funders. We've got a mix of both biomedical and optics students. I think it's a really great group, and everyone does amazing work. And so just, again, kind of going back to the overview thing, if there's any questions that you guys have, I'd love to take them now. But really, the whole purpose of this is really trying to bring it back to healthcare and improve patient outcomes and patient lives. So thank you very much for your time.
So is the important information contained in the optical signal? Can't you probe, for example, the small changes in local temperature? If cells are actively doing something, maybe the local temperature varies by a fraction of a degree. Or the softness of the tissue, you can just touch it and see if the tissue is softer or harder than it should be. So focusing on optics, aren't you missing some other things that might be relevant as well?
That's a great point, and specifically with the elasticity of the tissue, a very common diagnostic technique is literally to go in there and palpitate the tissue and be like, oh, is it hard or is it soft? And so clearly the stiffness has something to do with it. To some extent, these things are all sort of intertwined. And so, for example, if you're looking at like the collagen abundance and how randomized it is, that will lead to stiffer tissues. There are optical modalities to measure that. For example, you can do what's called elastography, where you essentially do an interferometric technique to see how much things have moved, and if you measure the force you applied, you can then calculate the stiffness. In terms of temperature, that's a very good point. I think the optoacoustic effect would probably be the most relevant for trying to measure variations in temperature. I don't know the boundaries of sensitivity on that, though. So I'm not sure if a fraction of a degree is possible using current methods. But I agree with you that taking kind of a multimodal approach to try and introduce things like stiffness measurements or temperature measurements could augment these as well. I think it very much depends on the disease and what the characteristics of that specific disease are.
But it doesn't have to be optical. You can use a thermometer, for example, a sensitive thermometer to measure the temperature. The probe is there. You could just touch the esophagus and measure the temperature.
Yeah, that's a great point as well. That especially with the idea of like an added probe. And so when we look at how these endoscopes are designed, they typically do have an extra channel. And so here I'm introducing my little scope to it, but we could equivalently just pull that out and introduce a thermometer as well. Or we could have yet another working channel in my device, which then lets you do a temperature measurement. So that's definitely possible. Any other question?
Yeah, so in the first section you had images where you compared like an optical image with compared to NADH and SHG. So you were talking about the metabolism. So what features are you looking at to see that there's more metabolism? And then you talked about the ratio of NADH to FAD. Is that the main parameter that you use to measure the metabolism?
Yeah, great question. Let me jump back to those slides. With these guys, or maybe actually let's go to these guys because this have FAD on there as well. Yeah, so one thing about imaging features is that whenever you use absolute values, it becomes tough because if you have variations in your illumination or your working distance, that kind of messes things up. So doing ratios is quite common. So the main measurement in terms of metabolism would both be the amount of NADH, which again is tough because you might have those day-to-day variations, but the ratio of NADH to FAD. And so that's referred to as the redox ratio, which refers to reduction oxidation. And specifically, again, if you have tissues which are undergoing what's called oxidative phosphorylation, that's what produces energy normally. They'll produce some NADH, but then if you have it, in the case of cancer, when you have what's called the Warburg effect and they don't have oxygen anymore, they start to go to aerobic glycolysis where they produce a lot more NADH. So you actually see that this rate, but the FAD stays the same. And that's the important thing, is that that's kind of a normalizing factor. And so you would expect that the ratio of these two increases with cancer, kind of suggesting that this Warburg effect is happening, where the tissue no longer has the oxygen to produce the metabolism, so it switches the way that it produces energy to something else.
Okay, so during the early stages of epithelial cancers, are there strong symptoms that the patient can feel? Because if not, why would someone go for early screening?
So for things like esophageal cancers, almost no symptoms. And so good question, why do people go for early screening? There are major risk factors. And so for example, if you have that condition, Barrett's esophagus, you have about 30-fold increased risk of getting esophageal cancer. And so they will go for routine screening. Unfortunately, just screening isn't very effective. They'll show up, but like I said, people just take random biopsies, and you can imagine that a small lesion is pretty small, so if you miss that, you're out of luck. Other things that are kind of risk factors which might cause you to go get screened is if you have what's called gastroesophageal reflux disease, as well as genetic markers. So we've probably all heard of BRCA mutations in breast cancer. So those like high-risk populations are those which could really benefit from increased screening. There are some cancers which do have symptoms early on, but really the ones that need better early screening are those that don't, because it's sort of a ghost until it's too late. And that is quite common with GI cancers, so things in the small intestine or the esophagus. But then a lot of my PhD work with Jennifer was on ovarian cancer, which also doesn't have any symptoms until you're essentially, it's metastasized to like your liver, and then at that point it's too late.
So are you trying to commercialize these?
I mean, that would be great. I think, to me, if it improves people's lives and helps, and sometimes that's the fastest way. Like a big company that has tons of resources, if they are the ones to take a concept from development to production, that's the right way to do it. So I mean, I think, for me, it's not financial motivation, but definitely I do think that realistically is the best way to get something used in the clinic.
And are there big companies you're competing against?
Competing or working? I don't know about competing. I think that everyone sort of has their niches, and I feel like one thing with companies is that they typically won't buy into something until it's been proven pretty well, and so they don't really do a whole lot of their own like R&D. Smaller companies, I work with a couple of sort of smaller companies in town that do like endoscopes and stuff, but I'd say that's more of a collaborative thing. So definitely have some industry connections, but I wouldn't say that I'm working against them. I don't think I would win.
No, go ahead. Okay. I think it's very interesting, and I think a cool advantage with the hyperspectral imaging and the kind of three-dimensional voxels is you could maybe incorporate a type of in situ, more targeted photodynamic therapy for the cancerous cells. So you could go ahead and deliver the photosensitized drugs, and I don't know your thoughts on that.
Yeah, absolutely. I think I framed this whole thing in diagnosis, but there's also many different ways that this could be used as a tool to enhance other things. So when I talked a little bit about like this cell lineage tracking, this is very much a tool to increase understanding. It's not to help diagnose. And similarly, it could be used to guide therapeutics. Surgical guidance is also a really big area for this. And so I, again, didn't go too much into the details, but with this disease gastronoma, really the key is surgical guidance. It's not diagnosis. And so in this case, it would be something like helping improve therapies. Photodynamics is great, and if you can image through depth as well, which you can do with multi-photon. I'm not showing that here, but you can actually get a three-dimensional data set, or other modalities like optical coherence tomography. Those would be great for doing something like photodynamic therapy. And that's becoming more common with esophageal cancer, actually. You do have radiotherapy ablation and stuff.
So, how does thresholding work? Like, how do you... Because there were some pictures that I couldn't really see much difference.
Right.
So how does that work, and what's the chances, I guess, for false positive or misdetection?
That is also a great question. So it's done very carefully. I think normally you will do it in some way relative. So again, it's not an absolute number where you're like, oh, I'm going to threshold this at 10. Or just because if you have variations day to day, it might mess that up. So again, ratios are helpful. Other things that are helpful are looking at sort of the spatial characteristics and distributions of the values. And so, like, for example, the distribution of the values in this are going to be much different than this because we have a lot more black spots. So again, that's all relative, and so it makes it a lot easier to practically implement. In terms of so what you refer to as false positives and stuff, there's a bunch of different terms. There's sensitivity and specificity, false positive rate, false negative rate. So for any disease, this is actually very well developed. But basically, they have a certain threshold that you need to reach before it's feasible to implement clinically. And so for something like esophageal cancer, it needs to be about 90%. 90% specific and sensitive. So specificity is... Rather, sensitivity is essentially how much do you find the disease correctly. Specificity is how much do you not have false positives. And so for something like screening, you actually don't care that much about specificity, because what you would do is you would have a screening test. Oh, it looks like it might be dangerous. Then you go for a diagnostic test. So the diagnostic ones need to have very, very high specificity, because after that is when you go have surgery or something. And so it's this interesting paradigm that totally depends on what niche you're trying to fill. But something like esophageal, it needs to be about 90%. I can't really tell you what the results are for this image set because we're analyzing it right now, so we don't have it. When I was doing the ovarian stuff, that we got about 85-90% using OCT and multi-photon. So it does pretty well, but that is sort of right at the boundary. If it was 99, then there's no question that this is the best thing ever. But it does become quite challenging when you're kind of right at that threshold. Honestly, the whole concept of the economics of the healthcare system is interesting too, because in your device development, there's like a number that you have to reach in terms of the amount it has to cost to produce one before anyone's even going to care. And it has to do with how much people get reimbursed through insurance and stuff. And so for, again, esophageal, $190. So whatever you make has to be $190 or less, or it's not feasible for screening. And every disease is different because every procedure is different and all that sort of thing. But it's a very interesting topic that there's textbooks written on and stuff that it's fascinating in my opinion. But as you know, with research, it doesn't always necessarily have to be right at that point right away. Yeah, but if something is proven to be 99% specific and sensitive, then probably the work would be done to get it to that price point. Mm-hmm. Thank you.
Actually, I got two questions. First one is for the customer multi-spectral filter array.
Mm-hmm.
Will the crosstalk be a potential problem, especially when the two selected spectra are quite close to each other? And my second question is about the polarization. So I'm quite curious about what's the major polarization differences between benign tissue and cancer tissue. Is the differences, is it on the degree of polarization, or they change the polarization scale, for example, from linear to circular, and how strong this polarization differences we can see?
Yeah, no, those are both great questions. So crosstalk, essentially there will be some crosstalk, and it has to do with the F number of your system. And so if you could imagine that if you had a filter and the light was only coming in parallel to it, you're fine because it just goes straight through to the pixel. If you're focusing it, it obviously is coming in at an angle, and then you can have it sort of bleeding over to the other pixels. Sometimes you fabricate these things so the pixels have like little wells between them to reduce the crosstalk. And the other limit is essentially the bandwidth, that you're going to have some bleed over in terms of the bandwidth of the spectral shape. So the limits on that are 10 to 15 nanometers bandwidth. That's about as good as you can get. And then in terms of the actual bleeding over from pixel to pixel, if you take the approach of having some space between the pixels, it usually reduces that to at least the level of like the thermal noise in the system, so it's not a huge deal. That's a great question, though, because absolutely, you have to design your optics as well to be appropriate for this. If you have a super high NA system and it's all coming in really steeply, that causes a ton of crosstalk. That's hard to get rid of. So typically, you actually want to use telecentric systems for these things, which is kind of a pain, but it is what it is. And if you design custom optics for it, then it's a little bit easier to get around. But machine vision lenses and stuff are good for that. Does that answer your question, the first question? And so polarization, good question. So let's see, what is the best slide for this? The kind of three. Let's go here. The three polarization things that happen most in tissue are depolarizing. And so tissue is a very scattering medium, and so depolarization is quite high. And it's significantly higher in cancerous tissues. And so that's a pretty good measure. I'd say it's maybe 1.5 times higher in some studies. It's all studies are different. But then you have retardants. And so retardants can either be the sort of amount of retardants or the fast axis, how it's oriented. And so in particular, the one that's interesting is how it's oriented. And so you can see if you have a really high kind of resolution for mapping out connective tissue, like if you measure your muscle, that should have a really nicely oriented retardance fast axis and a high amount of retardance, because it essentially is just like a bunch of vertical structures. And so it very much depends on what type of tissue you're looking at. That if you have a very structured tissue, retardance becomes a very good measure of what you're looking at. Circular tends to not, especially for diagnostics, you don't really see much circular polarization effects. That only occurs if you're doing like brain imaging and you see glucose being hugely deposited around there, because glucose is chiral. But I would say that probably depolarization would be number one. Then number two would be looking at the retardance map and whether it sort of is organized or disorganized. But for some diseases, that would not be very helpful because, like for example, again, I'm talking a lot about the ovary, but that's just kind of a mass of tissue. There's no structure to it. It's just a mass, and so it has no retardance to it. Polarization imaging for that's not going to be quite very helpful. But for the esophagus, there is actually some structural nature to it, given that it's kind of a long tube. And so retardance can be a bit more useful in that context. Mm-hmm. Hey, machine learning question?
No. Actually, when you showed us the endoscope after you did the lithographic fabrication.
Yeah, the one at the end?
Yeah. Yeah, this one right here. What is the purpose of the lens array here?
So the micro lens array helps a bit with uniformity. It's kind of trying to, you're essentially trying to like collimate the fiber array, but you have a bunch of those things that are trying to collimate it, and so you overlap all of the different, all of the different projection images. And so micro lens arrays are typically just used for trying to enhance uniformity. You try and place the source at the focal point of that lens array, and it kind of goes in the opposite direction, that you can also put them on sensors to try and collect more light. And so this isn't particular. This is very much a schematic diagram. Like, this has not been designed such that the specifications of the microlens array will produce 80% uniformity or whatever. But it's just a tool that's used a lot for when you have non-uniform sources. Like, these fibers are super directional and non-uniform.
Is the fiber, so then the lights, is it addressable behind each microlens? So you could directionally control the light source?
It would not be directionally controlled. So that would kind of be bonded in there, so it's a bunch of fibers that are facing forward. And then the microlens array would go in front of it and just try and project light to the front. I think, theoretically you could put in some kind of mechanism to adjust the light, but at least in this design, there's no mechanism to do that. And that would be tough. And I think, again, it goes into the specs of like what's your field of view, what's your working distance, and you would want to try and make it such that the projection of the illumination is relatively uniform over that, in what you're interested in.
So is it like one fiber per lens in that case? Or maybe like 6 bundles per, because then you can just turn on one and then directionally like the same
top left one under each lens.
Oh, I see what you're saying.
Yeah.
Yeah. That's interesting. That's a good concept. I hadn't actually thought about that.
You could do Fourier tychography and do phase retrieval.
So I actually have thought about doing structured illumination through endoscopes and stuff, and I think that very much you could do with fibers in specific clever orientations, especially if they happen to be coherent with one another. Yeah. So yeah, definitely possible. I haven't looked into it in depth, but that's a really good idea. Yeah, sure. I'll leave it up to you.
So I guess spectral wavelengths that you're sampling for this multispectral filter are sort of informed by the end member signals, right?
Absolutely.
For each of these signatures.
Mm-hmm.
And I'm wondering if you're selecting those wavelengths based on like what are you optimizing over? Is it over differentiation between each of the end members, or is it over SNR? And isn't there kind of a trade-off there?
Yeah. Also, very good question. Yeah, so you have to also pick the merit function of like what is it that you care about most, and so some of that could be. Let me go to this guy again. Usually what I would be trying to do is estimate how closely we recover the end member abundance, and so that would be essentially like the RMS difference of each one. That you take the real one, your estimated one, take the difference, do that for all of them. Square them, add them up, square root, and then that's your merit function. And so if they're super close, that's going to be very low, and so you try and minimize it. Alternatively, you could look at things like sort of the resolution. The spatial resolution essentially could be a merit function, and that will be entirely defined by how big your mosaic is. And so it is a tough problem, and so there's like different types of merit functions you could choose, and I think the issue is that there's no right answer. In the end, it's very much driven by what your application is going to be. That if you really care about trying to classify like an object, then maybe you do care most about the end member abundance, because that's essentially classification. But if you're really trying to do something like in the middle, where you provide really good visual quality, for example, if you're trying to make a complete imager, not something that augments a normal endoscope, you might care just as much about the visual product, because then the person can also see what they're looking at, and a lot of times that is how they do diagnosis. But then you might want to balance and do sort of a merit function which has a little bit of both, contribution both from the resolution standpoint and from the unmixing standpoint. But yeah, I guess the short answer is there is no right answer, but we usually just do the accuracy of the estimated end members.